Evening, Dr. Moore. This is my presentation on integration of AI with CRISPR gene editing technology. Here we go. Introduction. Make sure you can see it. CRISPR and artificial intelligence AI are two of the most transformative technologies in modern science. CRISPR enables precise editing of DNA, offering groundbreaking possibilities in genetics, medicine, and biotechnology. At the same time, AI brings powerful data processing and pattern recognition capabilities that are essential for navigating the complexity of biological systems. When integrated, AI and CRISPR complement each other. AI enhances the precision, efficiency, and predictive power of CRISPR gene editing, while CRISPR provides the experimental data needed to train and refine AI models. This synergy is unlocking new frontiers in genome engineering, from personalized medicine to functional genomics. All right, what is CRISPR? That would be the cluster regularly interspaced short palindromic repeats gene editing tool that enables precise DNA modification. Also uses Cas9 protein guided by RNA to target specific gene sequences. So it's basically just gene sequences, altering them to not a mutation, but to better help, human beings. Some instances there's DNA sequences that are messed up because of mutations. So CRISPR, they go in and they'll use the technology to fix it. What is artificial intelligence? Computer systems simulating human intelligence, machine learning, neural networks, and deep learning can analyze vast biological data or CRISPR target. I'm sure you know what AI is, but artificial intelligence, nowadays in 2025, especially 2024, there was a lot going on about AI and everyone's talking about, the metaverse and things like that. So we integrate AI with CRISPR. It'll be able to allow our medical fields to, beat the times, get down with the technology, and also push an agenda that is beyond, people saying AI is going to take over the world. It'll show that AI is actually alternating the way that the future will, progress, help our medicine and our diseases. I won't say decrease because we can't make that guarantee, but help fight the battle. Why integrate AI with CRISPR? Enhances target accuracy and efficiency, predicts off-target effects, accelerates discovery and applications. Example 1, predicting off-target effects. One of the biggest risks in CRISPR gene editing, off-target cuts can lead to harmful mutations. Off-target effects occur when the CRISPR-Cas9 system cuts DNA at the unintended locations that are similar but not identical to the target sequences. These unintended edits can 1. Introduce mutations that disrupt other genes of regulatory regions. 2. Lead to chromosomal rearrangements. 3. Cause toxic effects or malignant transformations in cells, especially dangerous in clinical settings. Tools for off-target prediction. AI especially, I don't know, hopefully when you're on this slide, you look at the slides because this is pretty small, I'm sure. AI, especially machine learning, ML, and deep learning models has transformed how researchers predict and avoid off-target activity. These models learn patterns from large data sets and generalize them to unseen cases. Key approaches include 1. Deep learning models. Deep CRISPR, net, and elevation are examples of models that use neural networks trained on CRISPR editing datasets. These models evaluate a guide RNA's sequence along with the genomic context to score the likelihood of off-target. They consider features such as mismatch tolerance, base pair thermodynamics, DNA shape and bendability, epigenetic markers like histone modifications and DNA methylation. 2. Genome-wide off-target scanning. AI models can analyze entire genome to flag potential off-target regions. Just being able to scan, DNA sequences with AI, you'll be able to see these where pinpoint each mutation is instead of having to do what they knew back in the day and break it down, take hours, months. But with AI, you'll be able to break it down quicker, get down to where you need to, fix the mutation and then use CRISPR to flip it and fix it. Use heuristics to scan for sequences with up to 3 to 5 mismatches from the guide RNA. Prioritize results based on gene proximity, known disease associations, or chromosome state. Impact of off-target prediction. It reduces the unintended edits, improves safety for therapeutic applications, enables more responsible gene editing practices. Example 2, guide RNA design. Guide RNA is crucial for CRISPR accuracy. Poorly designed gRNAs reduce efficiency or cause errors. AI optimized gRNA design. What is gRNA and why is it important? The guide RNA is a CRISPR Cas9 system. It directs the Cas9 protein to the specific DNA sequence that needs to be edited. A well-designed gRNA ensures high on-target activity, efficient DNA cutting at the intended site, low off-target activity, avoiding unintended DNA modifications, greater specifically, especially in their therapeutic context where precision is critical. gRNA, I don't know, back in your day when you was in college, you probably went over genetics. gRNA is just a guide RNA. It's the first RNA in a DNA sequence. So, benefits AI-driven gRNA design: enhances precision and reproducibility, saves time in experimental design, customizes edits for specific cell types. Example 3, AI for disease-specific editing. AI identifies genetic mutations associated with diseases, guides CRISPR to edit disease-causing sequences. Case study sickle cell disease, for example. AI analyzed patient genomes to pinpoint ideal CRISPR targets. CRISPR corrected the mutation in hematopoietic stem cells. I'm going to quickly go over this slide. AI applications in genetic diseases. AI algorithms, especially those using deep learning, are trained on massive genomic data sets to identify mutations associated with various genetic disorders. AI helps design customized CRISPR interventions based on a patient's unique genetic profile by analyzing whole genome sequencing data. Example 4, AI-assisted functional genomics. Example 4, AI-assisted functional genomics. Overview. Functional genomics aims to understand the roles of genes and regulatory elements in biological procedures. AI enhances these efforts by analyzing the vast amount of resulting data to uncover gene functions, interactions, and potential in therapeutic targets. How AI enhances it. AI models detect subtle patterns in phenotypic responses across thousands of CRISPR perturbations. Machine learning clusters genes by shared biological functions and pathways based on CRISPR screen data. Deep learning is used to predict how gene networks respond to the specific perturbations, enabling insights into complex diseases. Real-world application. In cancer research, AI and CRISPR have been combined to identify synthetic lethal interactions, gene pairs where simultaneously disruption kills cancer cells but spares normal ones. AI-assisted CRISPR screens have helped uncover non-coding regulatory elements that drive oncogene expression. Ethical considerations. Dual use of AI in gene editing raises ethical concerns, potential misuse, or unintended consequences. Calls for regulation and transparency. Challenges and limitations. Data quality and quantity limitations. Many existing genomic datasets come from specific populations, often underrepresenting ethnic and genetic diversity. There's a lack of experimentally validated off-target editing data. Biases in AI models and off-target predictions is not always perfect. Computer is not always the best option. You should also get in the field and do the work yourself. Future directions. Integration with other omics data, real-time monitoring of CRISPR edits with AI, personalized medicine, and synthetic biology. My summary and my conclusion basically say the same thing but in different ways, so I'm just going to read the conclusion. In conclusion, interdisciplinary collaboration, innovation, and ethical oversight are essential to fully harness the benefits of AI CRISPR integration with responsible development and regulation. This powerful combination has the potential to revolutionize disease treatment, genetic engineering, and the future of medicine. These are my references. Thank you for having me.